Okay, hello everybody. I'm heading a department at the Institute of Cell Biology, National Academy of Sciences in Ukraine, in Lviv, western part of Ukraine. And in this work, we were searching for new biomarkers. And we used a unique approach, as for me. Do you want to search a marker when you have 2,000 different proteins or peptides? Of course no, because it's very difficult. And we proposed a new approach, just searching the markers in the material which is thrown away by everybody. So when you have a bioliquid, let's say blood or saliva or tears or whatever, urine, usually you pellet all the proteins with trichloroacetic acid, TCA, I will say in next, and analyze 150 different proteins. Of course, it's very difficult, and most of them have been already started, and they have been analyzed by proteomics and other approaches. What we did, we took the waste, so the material, the supernatant which was left after such precipitation, and then we pelleted this supernatant with acetone, and then soluted these proteins and subjected to HPLC SDS electrophoresis. We cut it off a band from electrophoregram and subjected it to mass spectrometry and bioactivity study. And we found, as a result, when we took for analysis the blood serum of multiple sclerosis patients, we found that some unique proteins are present there which shouldn't be there. One of them is a protein which is everywhere, it's albumin, and another one was a unique one. It was a fragment of unconventional myosin, and it was a 48 kilodalton fragment. So we found that in patients with multiple sclerosis, most of them have higher or lower amount of this protein, and the same was done in systemic lupus erythematosus, again increased amount of this fragment. Somehow it was increased in rheumatoid arthritis patients. Then it was also increased in Alzheimer's disease patients, and it was also increased in non-Hodgkin lymphoma in blood serum from non-Hodgkin lymphoma patients. You can see this. While it was not so frequent in multiple myeloma, so this approach can be used for diagnosis, for prognosis. And besides these proteins, we also found that in this material we can also find some peptides. These peptides, they possess biological activities, their own biological activities. They are toxic for some tumor cells, and of course it's not so easy to work with peptides. So we put this, immobilize this peptide on the nanoparticles or polymeric nanoparticles, and found enhancement of their biological activity. So this original approach was also approved not only for blood serum, it was tested for urine. We found some unique proteins in urine using this approach which I described at the very beginning. Some of these data have been published in biochemical and biophysical reports and another protein and peptide letters, and it was also published in the monograph Myosin, Biosynthesis, Classes and Function. So if anybody interested in applying this approach, you're welcome to address us, and the email is available. Thank you.